Welcome to the 101st season of Watson Lectures at Caltech. I hope you all got a chance to enjoy some of the activities outside, some of the music, have a chance to have some exciting pre-science conversations. Maybe you got a chance to talk to our speaker, Richard Andersen's researchers, about the work that they're doing and that you're going to hear about tonight. And maybe, if you're lucky, you got a chance to speak with some of the participants in their research on brain-machine interfaces. Those participants, our guests of honor, are here in the front row. I also want to do a special welcome to the students in the audience. So we have students from Francisco Bravo Medical Magnet School in Lincoln Heights. Come on, you know who you are. They're part of our Junior Watson program. So they have a chance to tour Dr. Andersen's lab and learn a little bit about studying science and engineering here at Caltech. So who am I? I am Crystal Dilworth. I'm a science communicator, and I am your host for the evening. I'm also a Caltech alum, and I did my PhD in molecular neuroscience in the lab of Henry A. Lester, where I spent about 5.5 years making neurons glow in the dark. Since then, I've built a career making science accessible, exciting, and fun for the general public. And the Watsons offer an incredible opportunity to do just that. You have a chance to hear directly from the researchers that are furthering scientific innovation. So last month, we talked a little bit about how the Watsons got started. One man, Ernest C. Watson, with a mission of bringing the science that was happening on the Caltech campus to the people of Pasadena. So I thought maybe today we would hear a little bit from Dr. Watson himself. So in a letter that he wrote to the lecture committee in 1964, in an attempt to bring his lecture series here to the newly constructed Beckman Auditorium, he writes, "So let me close very much as I began, with another question. You say this is very interesting, and it's quite entertaining, but after all, what use is it? What good is it all? That question always annoys me. I don't see why you're willing to accept music, art, literature, even sports for their own sakes. But you always expect everything scientific to have an immediate practical application. Of course, it usually does. That's one of the satisfying things about scientific work. But the man who is only interested in the practical applications is ultimate justification of work of this kind. The real reason that we do it is not because of the practical application, but simply because it's fun." Now, the research that you're going to hear about that's being done by Richard Andersen has very important and immediate practical applications. And in a time when science and scientists are up against public mistrust and the proliferation of misinformation, it makes moments like these, lectures like these, exceptionally important. I mean, like I said, you get to hear directly from the people that are advancing scientific innovation, and you get to ask them questions. So, what can you expect from tonight? Well, before we hear about the science and his talk, Unlocking Movement, we're going to hear a little bit about Richard Andersen, the person. So you hear an introduction from the division chair in biology and biological engineering, and then there'll be a short video about Dr. Andersen's life and his work. Afterwards, that's when you guys get to ask your questions. So I don't know if there was a QR code here, but we're using the app called Slido. So if you haven't already downloaded it, please do so. You can download Slido, and that is how you will submit your questions to me. And then I will ask, hopefully all of them, to Dr. Andersen and the QA at the end. And then in conclusion, we got tea and coffee for you outside. a little bit more conversation about what you've heard about, and hopefully you've had some fun. All right, so let's get this show on the road. Please take the opportunity to silence your cell phones, your watches, and all of your tech that beeps, and take a look at your closest emergency exit, and allow me to welcome to the stage the chair of the Division of Biology and Biological Engineering, Dr. Richard Murray.
Thanks, Crystal. Good evening. As Crystal said, I'm Richard Murray. I'm the chair of the Division of Biology and Biological Engineering here at Caltech, and it's my honor to introduce tonight's speaker, Richard Andersen. Richard Andersen is the James G. Boswell Professor of Neuroscience here at Caltech. He spent a lot of his time moving back and forth across the country. He was born in Pennsylvania, but he did his undergraduate work at UC Davis, majoring in biochemistry, and then went to UCSF for his PhD in physiology. Then moved back to the East Coast to complete a postdoc at Johns Hopkins University in Baltimore, before taking on an assistant professorship position at the Salk Institute and UC San Diego, back on the West Coast. From there, was back to the East Coast to become an assistant professor at MIT, before finally returning to California about 30 years ago, where we've managed to keep him put since. And so we're very pleased that we've managed to keep Richard from that migration back and forth across the country. Richard has received numerous accolades for his research. He's a member of both the National Academy of Sciences and the National Academy of Medicine, as well as a fellow of the American Academy of Arts and Sciences. Richard gets to work on some of the most fascinating parts of biology. How does the brain work? As you might imagine, the brain's a pretty complicated organ, about 100 billion neurons, all working together to somehow allow people to do the amazing things that we do. Figuring out what's going on in the brain is really hard, because each of those 100 million or 100 billion neurons is itself has millions of molecules inside, and itself is a complex machine that's doing an enormous number of things. And so figuring out, right, how do we measure what's going on, how do we sort out what's going on, right, inside this complicated machine that's our brain is an enormous challenge. And it's this area that Richard's focused his career, bringing an array of new technologies and techniques to help us untangle some of the complexity of how we plan and execute motions. And we're going to hear more about that tonight. Richard's work is an excellent fit for the Division of Biology and Biological Engineering here. He, as Crystal said, works to, and Dr. Watson said, works to understand the basic science behind what's going on in our heads, but then uses that understanding to help develop technologies that can help humans to do things, right, when their brains or other body damage doesn't allow them to do that. And so when some of those functions have been lost. I've had the pleasure of working with Richard through a joint student in our labs, and it's just completely amazing the types of things that he and his lab mates are able to do. So it really is a pleasure to get to introduce him. And so with that, let me turn the stage over to Richard, but first we'll watch a short video that gives us an introduction to him. Thank you.
I grew up all over the place. My father was a chemical engineer. He'd talk to me about his work, sometimes a bit over my head, but I got the gist of it. There wasn't neuroscience at that time, so I kind of pieced together various classes, graduated in biochemistries, and worked in a lab recording from retinal ganglion cells in the visual system. So that was my first real research experience in neuroscience. I'm Richard Andersen. I'm a neuroscientist. I work here at Caltech on how the brain processes sensory information and transforms it into commands to make movements. My father had polio during World War II and later had post-polio syndrome, so was not very ambulatory. So I can relate to that. In the last few years, we've been able to actually do work in humans, helping people with tetraplegia so that they can someday be able to control their environment just with their thoughts. So we have a couple labs here at Caltech, so we do have several setups for human studies. For some of our subjects, we have equipment in their home. We hope to get to the position where it doesn't require a student visiting them to run the experiment. Not that they necessarily want to run experiments, but that they could use the devices to help them in their activities of daily living. We actually have a driving demonstration that we've done in collaboration with Ford, so they can drive a car from here in Los Angeles that's actually located in Michigan. Working with humans is enormously satisfying. They're the heroes in this field. They work more or less every week. It's like a job, it has a goal. I think that really improves the quality of life of the subjects. And then the reciprocal is they improve the quality of research in our life. So it's very different. It has so many dimensions when you work with humans. So it's great. Well, the great mysteries of the brain, if you talk to many neuroscientists, is consciousness. And in a way, we're beginning to study that, because when a subject is aware of what they intend to do, a lot of what's going on there is what we call intrinsic, which means it's below consciousness. But it can then bubble up into consciousness. So even if in the end never really understand it, it'll lead to a lot of findings about components of it. It's very wide open and very exciting. Hello, thank you for the nice introduction, Richard. Thank you, everyone, for coming tonight. Hopefully you had some of you were able to participate in the pre-show. I'd like to thank my students and the participants who manned the booths and talked to you about our science. So I'll be talking about unlocking movement, helping paralyzed people use thought to control computers and robots. And so this is just an outline of the talk. And first I'll say what a neuroprosthetic is. It's a brain-machine interface, and it's a device that can read out the intent of a subject. Then next I'll discuss cognitive neuroprosthetics. These are prosthetics, in principle, a lot of the recordings coming from posterior parietal cortex. And I'll give two examples of high cognitive signals we can decode there. One is social observation, and the other is silent language. Then we'll turn to actually writing signals into the brain. This is done to restore touch sensation, and this improves then the movement for control of a device like a robot hand. And then also I'll discuss a couple of new technologies that we've been developing along the way. One of them is a less invasive form of a brain-machine interface that uses ultrasound. And the second is the ability to drive an automobile by a paralyzed participant. So just to begin with, we have kind of two stages to a neuroprosthetic. So one of them is a readout, and we record neurons from the brain. It goes to a decoder, which uses machine learning to decipher what the activity means, and then sends a control signal to assistive devices like robots and computers. Now, we can also write in signals, and that has to do with writing in the sensation of touch. So there are many applications that the technology I'll be talking about can help out paralyzed patients. This includes spinal cord injury, stroke, MS, ALS, and Duchenne's muscular dystrophy. We've been concentrating on spinal cord injury, and in particular, high-level injuries up in the cervical areas. And so as a result, the subjects are tetraplegic. So what this means is they can't move any of their limbs or feel sensation from those limbs. And they have require generally round-the-clock assistive care from a caregiver. So our participants are the real heroes of this research, much like in that tape, that video that Peter made. And some of them are here tonight. They do this research with no benefit to themselves in curing paralysis. They do it rather out of the goodness of their heart for the generation of people after them once the technology is more refined. So this shows how the paralysis occurs. Normally, if you go to reach for an object, it comes in through the visual system. That projects up to visual cortex, which then proceeds to posterior parietal cortex, and then over to motor cortex, which sends motor neurons down the spinal cord. Those synapses on motor neurons in the spinal cord that then go out to the muscles to activate the limb. Now, this loop gets broken with a spinal cord lesion. So a subject can now no longer move their limb, but they can still think about moving. And so what we do is we being the neurosurgeons we work with, implant a small chip that can record the activity of brain cells. And the type of activity that we record is called a spike or an action potential. Is when a neuron fires, it generates, the cell body depolarizes, sends the signal down the axon to other neurons to influence them. And so this little graphic here shows that we actually don't put in a single electrode, but rather a whole array of electrodes. Each chip has 96 electrodes, and this little cartoon here shows about the scale of the recording. So you see the firing of neurons and the electrodes. So the neurons are very small, as are the electrodes. Now, next I'm going to show that we can amplify those signals by putting a speaker to the amplified signal, and we can actually hear the single neuron's activity. And that's one way we get a sense of what a neuron is doing. So you'll hear a kind of popping, so don't rush toward the exits, okay? And so that's what, and you'll be seeing more of this. This is what a single neuron looks like when it's firing and is being measured electrically. So these implants, and it's kind of amazing, they're very small. So here's one, it says 96 electrodes, and it's on the thumbnail there. It's 4 by 4 millimeters. And yet we can get an amazing amount of information, which we'll talk about later, from such a small recording device. And it's connected to wires that then go to a pedestal that's secured to the skull. And then each session, an amplifier is connected to that connector. So this shows an implant about the scale that it would have on a gyral surface within posterior parietal cortex. And what we look at is we look at the activity of the group of neurons. So we can see on the screen the firing of cells. We've highlighted one of them here. So again, you can see that sort of popping action potentials that we record. So this is a simulation of what a neuron might look like that is tuned. And the tuning is really essential for these experiments because the decoders have to interpret a pattern activity of cells that are tuned to different things. So in this example, neuron 1 is active for left movements, but not for right movements. You'll see a couple of figures which have a what we call a post-stimulus time histogram, which is the average of the activity. This blue area is the standard error for summing, in this case, 4 trials. Moving right, this simulated cell is not active. And each cell, or different cells have different tuning. So here's an example of a neuron that's active for right movement, but not left movement. So the real magic comes, or one of the magic points is how do you interpret this activity from a population of neurons? And we do that with a decoder. And the decoder takes in that activity that we saw on the previous slide, and it extracts features and then uses model fitting with machine learning to interpret that activity. So we use the spiking, as I showed you, and we have several neurons. They have their activity come in. We count the spikes. We use that information to then train a model that interprets that pattern of activity. Once it's trained, then on a trial, we get a pattern of activity in, and based on the spiking of those different neurons, the model predicts what the subject wants to do. So in this case, it would be to move a cursor down and to the left. So there will be 3 areas of the cortex that we'll be interested in, and this is different areas of the cortex doing different things is the concept behind cognitive neural prosthetics. So the idea is you go to an area of the brain that produces a particular function, and that's, say, if you want to go to recover speech, you go to a speech area, limb movement to a limb movement area. Early on, it was thought that the brain was so plastic that you could implant one of these probes anywhere and just learn to use it. But it turns out, after paralysis in adult, the structure of the brain has become rather crystallized. And so to get the signals you want, you can utilize the intrinsic structure that already exists within the brain. So the areas that we'll be interested in, one is the motor cortex, and this codes a trajectory for movement. And this is where most of the groups that do this kind of work make their implants. Now, somatosensory cortex is the area that responds when you feel something, and we'll be showing you later that we can stimulate that area to write in again the feeling of touch. And then the posterior parietal cortex is an area that's very high level or very cognitive, and it's an area that we've studied for a while, and one of the areas that our lab concentrates on. So the posterior parietal cortex, it's referred to by people in neuroscience as an association area because it associates the activity from many other regions of the brain. So it's connected to a large number of brains, and this particular region is involved in sensory to motor transformations. So in studies like imaging studies or studies of lesions and deficits that occur with stroke, it's been attributed to have many functions. These include action planning, visual processing, grasping and reaching eye movements, also tactile sensation, language processing and navigation, and actually many more things. So it's quite a heterogeneous place, processing a lot of information about both sensory and motor aspects of movement. So this is an example now of participant JJ, who's here today, who is controlling a cursor on a computer screen. So you can see when the target turns red, he moves the cursor to that location, and then back because the center one turns red again. And you can see that it's amazingly good. It's as good as I could do with a similar task. Now, we can also use these signals to control a robot. And this is participant EGS. And he wanted to be able to drink a beer, not having his mother help him. So this is the first time he was able to drink a beer on his own after his injury.
Very good. I'll pretend to drink my beer. All right. Cheers.
All right.
Hey, you finished that thing off. That was good.
Tyson says, drinking buddy.
And that's how science is
There it
made.
is. Look at this.
All right.
There you go.
So we were, Eric was very thrilled, as were we. This was a wonderful moment in time. So as you might expect then, this posterior parietal cortex, we can decode many different kinds of functions. So I showed you already that we could decode the goal and the trajectory, and also we can control all the fingers and actually all of the body, so leg movements as well as intended arm movements. I'll show you an example of action observation, which is important for social interaction, and also an example of silent language. That's the sort of inner dialogue that you have with yourself. So there are many things of importance to neuroprosthetics that we can decode from this very high-level cognitive area. So it's maybe not too surprising that we can decode all of that, but for me and my colleagues, what was the real mystery was, remember I showed you how tiny that array is. And so how is it that we can record from just a few hundred neurons with this tiny little patch of the brain and get out this much information? So this is an example of social observation, and as we'll see, this is possible because the neurons respond to more than one thing, but in different ways. And so you can pack a lot of information into a small number of cells. So in this case, what you'll see is that you'll hear the neuron firing when Nancy has her chin rubbed, but not her cheek. But the cell will also fire when Tyson, the experimenter, has his chin rubbed and not his cheek. So it's as if the cell in Nancy's brain is inferring what Tyson is feeling. So that's Now Tyson's asking Nancy to close her eyes. And so you can see that it's touch and not vision that the cell is active for, and that she's imagining perhaps that Tyson is feeling as well. So this we wanted to look at in much more detail, and so we looked at activity for an observer and someone who is actually touched. And we also looked at two body parts, in this case the cheek and the chin, or shoulder, I'm sorry, and 4 types of touch. So this is pinch and rub and tap and press. And so with all these variables then, this is one neuron. It shows that kind of histogram I mentioned earlier, and it responds to actual cheek and observed cheek, but not to the actual shoulder and observed shoulder. So this is a bit like the neuron that you saw in the movie. But if we look more carefully and we look at the different types of touch, what we find is that in the case of the actual cheek, it only responds then to the pinch, whereas for the observed cheek, it responds to all 4 types of touch. So things are actually mixed together, and this is probably one of the simpler neurons. So this mixing of information is quite profound, and we refer to that as mixed selectivity. Now, this is kind of just a toy model to explain how so many functions can be in one area. So we have here a plot of firing rate of 3 neurons along the 3 axes. And you can imagine that for a actual rub on the shoulder, that neuron 2 is much more active than the other 2. But for the actual rub on the cheek, here it's a different pattern of activity. And then if we look at the activity for observing these 2 types of action, we may see, again, a different pattern of activity in the 3 neurons. And so what this forms is what we call a subspace. And this might be a subspace for rubbing. And then we have another subspace for pinching. And then if we rotate these neurons into a representation in different coordinates that includes other cells, and these axes now have person, touch type, and body part along the axes, then we can fill in also the body part and the person. So when you have these different subspaces, then you can actually build, they're building blocks, and so you can build a multitude of different types of representations. So for instance, at the intersection here of observed and pinch and cheek, those you can construct that particular function. So this is a way that in a small group of neurons you could represent many, many different functions. Now, not all the posterior parietal cortex is one area with mixed neurons. They're actually different functional areas with different mixtures of variables. And all I've showed you so far is recordings from what's called the superior parietal lobule. And recently we put an array in the inferior parietal lobule, which had been implicated in grasping and in speech. And there we find actually activity related to when the subject is speaking. And this would be important for things like subjects that can't move their articulators anymore. That could come from being locked in from a stroke or from being paralyzed with ALS. So it'd be really nice, it would be really important to be able to read out their speech thoughts so they could still communicate. So this is a kind of a hot area right now. You might have seen it in the press. there are groups that have been able to, in the prefrontal cortex, read out actual speech, or even miming speech, in which they move the articulators but don't actually make a sound. But still, it has to be motor. It's been very difficult to find that sort of inner dialogue that you have, the speech that you have going on in your head that you're not actually speaking. So we actually have found that within the posterior parietal cortex. And we don't think that we're reading out people's internal thoughts, but rather their thoughts of or imagination of speech in the task. So this is what the subject sees on the screen. There's a word. They read the word. Then there's the cue, an orange cue, and they say the word internally. And then we decode what we think they're thinking, the word they're thinking, and feed it back to them. If it's green, that indicates we decoded it correctly. If it's red, we made a mistake. And so we used 8 words, 6 lexical words, and 2 pseudo words. And this shows 3 sessions, and you can see that it's very high performance using this, decoding this internal speech. So this is an example then. There's actually audio here, but of course, since FG, the subject, is not actually speaking, you don't hear anything. And so first he sees the cue to think about or read swimming, and then he gets the cue to say it to himself. Oops. And something went wrong there. So swimming, he gets the internal speech cue and gets swimming. And here are then 3 more words. So we decode battlefield correctly. Then cowboy, we get wrong. And finally, telephone, we get correctly. Whoops. Okay. So next we're going to shift away from reading out signals from the brain to actually writing them into the brain. And why is that important? Well, when you reach to grasp something, you use vision, but once you grasp it, to manipulate it, you need touch. And so this is, I think, a very robust example of that. This is an intact person striking a match. And this is once the fingertips have been anesthetized. You see, it can be very frustrating. So this is very important because if someone, again, as we mentioned, a problem with, besides spinal cord lesion blocking the motor signals out to the body, it also blocks the sensory pathways going back to the brain and the somatosensory cortex for feeling. So what we do in that case is we put touch sensors in the robotic hand, and then we can stimulate the somatosensory cortex with tiny currents and recreate the perception of touch. So this is from 2 arrays. So these are arrays just like we had for recording, But now we pass just a very tiny amount of current to stimulate a few neurons around the electrode tip. And the upper array, array 1, where we color in the boxes, those are the particular electrodes. And so you can see that for that array, it likes middle finger and ring finger. So the subject's feeling touch in those fingers again. And for the second array, it's for index and middle finger largely. And this recapitulates the map that's in intact subjects, a so-called somatotopic map of the body in the cortex. So this is participant AN, and he's here tonight. And what you'll see is that with brain control, he'll be controlling the finger on this virtual hand and touching a little blue object. And when he touches that, it'll then pass a little bit of current to that location on his fingertip, and he'll feel the touch. And in this, if you look at the arrays here, you can see the activity, and you can see the stimulation in these two right panels when the stimulation occurs. So now we'll put the two together, the stimulation along with the brain control, so that we'll have a complete feedback loop to improve dexterity with the robotic limb. Now, we've also developed a couple of new technologies along the way. One of them is the way that we can listen to the brain's activity with ultrasound. And you might wonder how you can do that. Well, when there's a stimulus that increases the metabolism that then causes the blood flow to increase. And when that happens, you can detect that with ultrasound recording. And there are a couple of really nice features to this type of device for brain-machine interfaces. And one is that it has a large field of view. So the electrodes that we have, you have to have the neuron in 150 microns to read the signal. And you saw it's a 4 by 4 millimeters, so it's a tiny little bit of cortex that you're sampling. With the ultrasound, you can see here that one section's been pulled out. And you can see that in this, it's not trying to optimize it, but here we can even go down 20 millimeters. This shows the section here through the intraparietal sulcus. Now, another feature that's nice is that it's less invasive. So if the ultrasound can't permeate the skull, but if you put a little window in it by surgically removing a little piece of the skull and replacing it with a transparent ultrasound window, then you can look into the brain. That contrasts with the surgery that you need to do to put in one of our arrays. There you have to not only go through the skull, but also the dura, and then push the array into the brain. So we've taken advantage of, well, it's no advantage, but it's there. With traumatic brain injury, there's a hemicraniotomy that's performed to relieve the pressure from the damage to the brain so that there isn't more damage from the pressure. And so a few months later then, a colleague that we collaborate with, Charles Liu, replaced that area of the skull that was removed with a material that's transparent to ultrasound. And J.H., who's in the audience tonight, this shows his actual surgery where this new material is placed. And so here's an example then looking down through the window. You can see the vasculature of the brain. This is in a case where nothing is happening. He's not doing anything. And then when he plays guitar, you see these areas light up with the activation. And you can see that very clearly through the window. We have three areas, small areas of interest we're looking at, and the gray is when he's playing guitar. And you can see two of them have really robust activations during guitar playing, but not during rest. Now, another technology that we're working on, and this is with Ford Motor Company and with BlackRock, is to be able to, for a tetraplegic person to be able to drive a car using brain control. And this is work with JJ, who's here. And these are simulations here. So one thing that was of high concern was whether it would be fast enough to brake. That seems pretty obvious. So you can see there is, I think it's a vending machine that suddenly appears in the road. And surprisingly, James can brake faster than age-matched controls. And the reason for that is the signals are coming directly out of the brain, so it doesn't have to go through the rest of the body. And this next clip is James is explaining why it's important to drive, which I imagine is certainly true in LA, but probably true everywhere.
Are you trying to stay in the lane, or?
Yeah, I'm trying to stabilize it. We're doing pretty good. Getting behind the wheel again, actually sitting in the front seat, in the driver's seat, that's gonna be pretty awesome because it's been almost 5 years now. And 5 years of sitting in the back seat, being at the mercy of my teenage son driving, it'd be great to finally have some control for myself.
So what we plan to do now, and Richard Murray mentioned we're with shared students are collaborating on semi-autonomous vehicles for paralyzed people. And the idea here is to use signals that, remember, we said are high level in posterior parietal cortex. There's navigation signals, there's emergency stop signals, which we just saw. There's also prediction and error correction. All those signals we find in the posterior parietal cortex. So the idea is then to use an AI controller to drive a semi-autonomous vehicle. So where do we go from here? Well, I think the path is pretty clear. We'll extend this cognitive neuroprosthetic concept to other areas of the brain, so particularly other high-level association areas. We'll also examine this idea of compositional coding and see if that holds for other brain areas. And then, we'll use, we saw with the language, the silent language, we can use now large language models. These are like tools that are used in the AI chat boxes you've heard so much about to begin to extract phonemes, whole words, and whole sentences from just language thought alone. Then on the development of technology side, we're continuing to optimize the ultrasound BMI, and we're also, of course, working on this prediction and error correction for driving. So in conclusion then, I'd really like to thank, again, the people that make this possible, our participants. Also, funding agencies, the Chen Brain Machine Center here at Caltech, also grants from NIH, the Boswell Foundation, and BlackRock. We collaborate with a number of labs here at Caltech, This has really been a great experience. We also collaborate with USC Medical School, UCLA Medical School, recently Colorado Medical School, the two main rehab centers here in LA, Casa Clina and Rancho Los Amigos. And I should also mention that we work with the inventor of the functional ultrasound technique in Paris, Mikhail Tanter, and with the company he has there, Iconius. And now, of course, these are current members in the lab. You met probably many of them if you went to the pre-show, or I'm sure some will be around after the show too to discuss things with. And on individual slides, you saw some of the past members that worked on some of the projects that I covered here. So thanks a lot.
Thank you so much, Richard. All right, now it's time for you guys to ask your questions. And I have to say, you are the most Caltech audience I've ever seen. We're talking about reading brains, and we're talking about influencing sensory perception, and the audience is just like, yeah, we get that. Do that every day. I always look into brains, always. There's windows in all the brains that I see. All right, so make sure you use the app, and Richard, if you want to have a seat, I think we'll start with maybe, the most obvious question, which is, of course, when can I get one? What is the process of commercializing something like this and taking it from research to?
Yeah, obviously, eventually we want to have this available in the clinic. It works in the laboratory, as you could see. And so there's no scientific or engineering blockade that has to be overcome to make it happen. But it would require probably a lot of money because it needs to be wireless, it needs to go through clinical trials and testing, then also it needs to be covered by insurance and all these sorts of things. So I once, about 20 years ago, I was interviewed by Time magazine. They asked, when will you figure out the brain? And I said, oh, in about 10 years. And, and that was the last sentence in the article. And my wife said, never do that again. So I take the fifth here.
Okay, protecting yourself. I get it. So obviously, there's very practical application to this type of research where you're trying to mimic activities that are already known. But what about the use of this technology for enhancement?
Yeah, that's a big question, of course, that you see that in a lot of science fiction, predated all this work. And so for our particular applications, they're strictly medical. But people like to ask us about enhancement. We ourselves don't work on that at this point. But people speculate about that. It has a lot of ethical issues like, say, for instance, who gets the implants? They're going to cost money. Will rich people get it and not poor people? So anyway, yeah, it's kind of futuristic, kind of sci-fi. I kind of stay away from that a bit, even though it's fun to talk about over a beer or something.
Well, I have another one of those questions for you. Somebody in the audience wants to know if this is the next generation polygraph. So is this being used for interrogations yet? And when can we expect that military application?
Okay. So
how good are we at reading words?
Yeah. So this is, again, a question of can we read someone's thoughts? And so originally they tried that with MRI. And the idea there was that you could tell when someone was lying. And this would be like the Stroop effect where you have conflict. And so it would show up in particular known parts of the brain. We actually tried that many years ago and it worked, but then we, of course, didn't pursue it. But it turns out a lot of people did. One person had a company that I'm aware of in the Bay Area, and it turned out that people could block it. So, and I think the same is going to be true with the implants and the language. I think if they're involved in a task and they're asked to say the word internally for you to hear it, but I think if they don't want you to hear something, just like maybe you're thinking something you wouldn't want someone to hear, I think it'll be able to do the same thing.
What amount of brain development is necessary before this technology becomes relevant? There's a question here about, would it work on someone that was born paralyzed, or do those connections need to be formed first before they can be decoded?
Yes, that would be a good question. We've only made these implants in adults, and it was actually, we expected that there would have been much more, especially some of our subjects were 10 years out from paralysis, that the cortex might have reorganized in such a way that it would make this technology difficult. But it turned out it didn't. And I think, though, early on during critical periods where your children are developing, you just don't have enough genes to take into account everything around you. So you have critical periods where you learn based on the environment, but then that tends to shut down after that. So I think we're beyond most critical periods, at least for adults.
Since this is used to decode thoughts for speech, what thoughts are allowed to come forward? Like, how are thoughts filtered?
Good question. We haven't tried that yet. But we've thought about it. We've thought, if someone kind of clears their mind, what that would look like. Presumably, we couldn't decode anything. Then also, we would try things like blocking it, as you mentioned. Yeah.
I mean, obviously, this is really, I'm a neuroscientist, I can say this. Obviously, this is really cool.
Thank you.
But how did you get started in this type of research?
Well, it kind of filtered in from animal work we did. And well, actually, initially, when I was a graduate student, I went to work with someone at UCSF who was working with a neurosurgeon who was looking at the awareness of intent in humans. I thought this was great. Unfortunately, the neurosurgeon got cancer and died. I had to change labs. And it wasn't until many years later that we actually did the same experiment and looked at awareness of intent. So it's kind of been a long cycle, but got there.
Yeah, it's always funny how you never quite end up where you think you're going to when you're following a line of research.
I think back then it wasn't practical. There weren't the tools like this. Population recording is really necessary to do this kind of brain control and readout of what people are thinking. The same is true for the microstimulation too, because you need very small currents. If you use big currents, you just feel tingling and kind of like a shock, like you grabbed an electric wire. But in our subjects, they feel things like pressure or rubbing or something blowing or even the feeling of their limb moving. So it becomes very precise when it's very small.
How long does an implant last?
Good question. Originally, we thought they'd only last about a year from animal studies, but generally we're finding they last about 5 years. One of our participants is now going into his 8th year, so this is a good thing. Still, it means that we'd like it to last a lifetime, so it's at this point it would require reimplantation. The good news is where we have explanted, once the arrays have stopped working properly and looking at them with scanning electron microscopy, It's just materials defects. So the insulation cracks, the tips fall off, things like that. So it's not an immune response, which would have been much harder to solve. It's a materials problem.
So when you're dealing with this additional layer in your research, which is really having a partnership with, the participants, look, with animal work, like mice don't talk back, but they also can't tell you where it hurts. So can you maybe speak to that additional element of data and the personal connection with your participants?
Yeah, it's fabulous. From my side, I can see how our research can be translated immediately. From a social side, it's wonderful to work with the participants, and it's kind of a lifelong bond because you get to know their families, they get to know you, the students, they see us all 2 or 3 times a week, if they can stand us. And that lasts many years, and then post-study, we keep in touch and also interact, ask them as consultants, things like that.
Well, thank you very much for being here and sharing your research with us. Can we get another round of applause for Dr. Andersen?
Thank you.
Well, thank you all for being here and for asking your questions. And like I said, please join us for tea and coffee outside. Good night.